Today we're going to cover considerations, challenges, and possible solutions for scalable manufacturing of MSC-derived EVs. I'm Taby Ahsan, Vice President of Research and Development at RoosterBio. As many of you know, mesenchymal stem/stromal cells, or MSCs, are used in a whole host of regenerative medicine applications. At times they're used as regenerative drugs, or their immunomodulatory properties are leveraged, as in the case of graft versus host disease. At other times, they're the cell source for engineered tissues or organs, or the cell platform for genetic engineering therapies. Today, we're going to be focusing on extracellular vesicles, which are some of the MSC-derived materials. As you all know, EVs are currently being investigated in a whole host of clinical applications, from bone fracture to wound repair. In many cases, EVs are being investigated as a novel therapeutic in and of themselves. In some other cases, they're being used as a simplified product design of something that has already been shown to be efficacious through MSC treatment. The EVs then act as the bioactive agent without the complexities of a viable product such as cells. Currently, the publication and clinical trial rate of MSC-EVs are mimicking that of MSCs. First, you start with basic research, you engage in clinical trials, and hopefully, ultimately, we end up with a commercial product. One of the aspects that MSCs and EVs can leverage is the well-vetted safety profile of MSCs. And therefore, that pipeline from basic research to commercial product may be accelerated for MSC-derived EVs. As you all know, the cells secrete the EVs. The EVs then are biologically active, containing all sorts of transmembrane proteins and cargo, such as RNA and proteins, that then, when transplanted in vivo, hope to have a mechanism of action that leads to efficacious treatment of pathologies. But what we don't know, and as many of the talks at this conference engage in, is we don't have a thorough understanding of EV biogenesis. We also don't have strong analytical tools to develop high-quality, critical quality attributes, or CQAs, of EV populations. And finally, we still are struggling to understand, in many cases, the mechanism of action upon transplantation. And as a result, because of this stage where we are still in the early stages of understanding EVs and their critical quality attributes and their mechanisms of action, the process by which we generate EVs is critical to the product definition of EVs. So what is the process? Well, here I show the typical EV manufacturing process. There's upstream processing, then there's subsequent downstream processing through various types of filtrations or purification processes, and then we have the analytics. Others will discuss how the analytics are being developed and how downstream processes are also being developed. Here at RoosterBio, we focus on the upstream process and how we can maximize the cell yields in order to maximize EV yields. When we think about the upstream process for EV manufacturing from MSCs, we have to think about multiple aspects. We have to think about the tissue source, whether those MSCs are from the umbilical cord or adipose or bone marrow, whether the donor is allogeneic or autologous, of course, things like sex and gender, as well as the health state of the donor matter. We need to consider the medium composition, whether it contains animal sera or human plasmolysate. And what is the cell state of the MSC itself, whether it's undifferentiated or if it's differentiated or even primed in order to promote EV productivity. And then also the replicative state. We do know that primary cells like MSCs go through replicative senescence, and therefore it's very important to maintain a consistent product population doubling level. And then finally, the manufacturing platform, whether it is in 2D adherent cultures or in 3D suspension bioreactors. Today, I'm going to be focusing on bone marrow with a little bit of data related to umbilical cord. We'll be focused on allogeneic cells in media that is free of animal sera. We're going to be focusing on non-differentiated or undifferentiated MSCs and at a fixed replicative state. But in essence, we're going to be focusing at the differences between 2D and 3D culture, given those constraints. As we discussed, EVs must go through basic research, clinical trials before they become a commercial product. We already have some information on the dosing numbers from basic research projects, for which we expect that when we have a peak commercial demand, that we would need billions or trillions of cells in order to generate the EVs appropriate for commercial availability. But as I discussed before, the process is the product. And therefore, you want to minimize the changes to your process as you move from basic research to peak commercial demand. That way, you can maintain your efficacious product throughout the different stages of translation. In that case, you have to also think about the end in terms of how you work into your basic research. But now let's look at some of the numbers. A single dose from the literature seems to be around 500 million cells, or 0.5 e to the 12 EVs. If we translate that to how MSCs are manufactured in a GMP environment, we find that a single 10-layer cell stack is about 1 dose. Now, we know that the key to a high EV yield is high volumes of cells. So how does this relate when we end up with a single dose from a 10-layer cell stack, which is currently the standard way to produce MSCs in a GMP environment? Well, let's dig into that a little bit more. So let's look at 2D adherent culture and the resources that required. If you have a 20-cell stack process that yields about 5 billion cells, it'll take about 10 hours. It'll require 200 square feet, and it will require 5 operators. If you then need to scale that up, all of those resources will almost scale linearly. Now remember, I mentioned that a single cell stack is about 1 dose. So if you want hundreds of doses per lot so that you can allow for testing and yet still have lot sizes that have some benefits of scale, then you now would need to create a process for hundreds of cell stacks. And you can see from the time, the square footage, and the operators required, that would become extremely resource intensive. Instead, if we think about another manufacturing platform, such as bioreactors, here in comparing a 60-cell stack to a 30-liter bioreactor, both yielding 15 billion cells, we can see that you save in time, you save in footprint, and you markedly save in operators. And this then becomes a scalable process, whereas the cell stacks are a scale-out process. So if we look at these manufacturing platforms, it is very easy to understand that as you go through log increases in the numbers of cells that you need at the various phases, from the lab scale to clinical trials, phase 1, 2, 3, to peak commercial demand, that your manufacturing platform necessarily must shift from the lab scale T-flasks to potentially multi-layer vessel production to suspension bioreactors. This will also allow you to benefit from the economy of scale as the prices per cell will decrease. And eventually, who knows, maybe we can drive these to cost only a few pennies per cell when we go to even larger bioreactor systems. So in this bioreactor system that is scalable, we used a classic growth and then starvation collection approach. So the cells initially expand in growth medium, but then through a wash and then switch to a media that is low particulate, we can then harvest EVs without any contamination from growth media components. And the process that we use is shown here on the right. We initially do a seed train that grows some cells on the adherent surfaces. We then inoculate the bioreactor at day 0. Midway through our culture, we add a feed. So this is a batch culture in the fact that we do not change our media, but we do add a feed in order to replenish some of the nutrients that get consumed during cell growth. At day 4 or 5, depending on the donor and the level of cell confluency, we then switch to a collection media. We then harvest EVs for 2 to 3 days. Now, we've used the scalable manufacturing platform already extensively to develop cell yields. And so we have a good understanding of the cell product formats that we need in order to start the seed train and eventually yield the cells coming out of the bioreactor. What we do not know is how this relates to EV productivity. And the importance in having this consistent CMC process as we go through scale will actually facilitate many regulatory hurdles as you advance in your translation from clinical trials to commercial product. So let us discuss how we end up developing those numbers of EV yield. Well, first let's look at cell expansion. Let's look at different scales. So here I've shown for both the 3-liter and the 15-liter, multiple runs, 2 runs at the 3-liter and 3 runs at the 15-liter scale, where I show cell density and as a reflection of cell growth over the culture duration. And then in the yellow shade, you see the collection phase. Now, when we switch to the collection media, in order to maintain a low particle, we've removed many of the mitogenic factors, and the cell numbers stay consistent and no longer are going through exponential growth. But that is when we begin to harvest our cells. And again, you can see the depiction on the right of the process. We inoculate the bioreactor, we allow it to grow for 4 days, we then switch to the collection media, and then we harvest the EVs. And so you can see that we end up with a very consistent trend in cell growth that's robust from time to time in this bioreactor. And if we look at the morphology at both the 3-liter and the 15-liter scale, these are microcarriers that have been coated with cells, and this is a typical cell expansion in microcarrier-laden cells in a suspension bioreactor. And on the right, you can see that we overlaid the 3-liter and the 15-liter bioreactor data for the cell numbers, and you can see again that this is very consistent as we scale up from the 3-liter to the 15-liter scale. Now, it's very important because the EVs, high-quality EVs, come from high-quality cells. It's very important that we characterize the MSCs at the end of the growth phase. And so we took them out in matched samples, and we looked at the trilineage differentiation properties, the cell surface markers, the angiogenic cytokine secretion, and the IDO activity of cells that had been grown in these 3D bioreactors. And we compared it to the 2D controls because the 2D culture is still the standard for MSCs. So as we shift to the bioreactor, we have to show comparability. And what we see is that in all of these properties, the cells are highly comparable between the 2D and 3D, indicating that the 3D bioreactor culture maintains the high quality CQAs of the MSC populations that are necessary to generate high quality EVs from MSCs. Subsequent to the cells and their growth phase, we then look at what are our EV collection density, or what our EV density is as over time, and what our yields are at the end of 3 days. So again, we did 2 runs in the 3-liter bioreactor and 3 runs in the 15-liter bioreactor. And you can see that our EV, or particle concentration, increases over time in a very consistent fashion. So this is a very reproducible process. And we end up with, on the order of, in the 15-liter bioreactor, around 3 to 4 e to the 12 EVs from each 15-liter process. And if we overlay those again, you can see that there is a difference in the EV productivity, which I will discuss more in a subsequent slide, in the 2D culture, which is in the dashed line, versus the bioreactor, which are the 2 solid lines. But the bioreactors at both the 3-liter and the 15-liter scale overlay, indicating that this is a highly scalable process and that it scales according to volume. But it is interesting to note that the EV productivity, or the EVs that are generated in the bioreactor system, are greater than they are in the 2D culture process. When we looked more carefully at the EVs that we end up with, in the top left, you can see that the EVs that are coming out of the bioreactor are of the same size as are expected from the 2D cultures as well. And these are all the metrics that are outlined in the ISEV guidelines. So the EV size is in the expected range. The cargo, the RNA cargo within the EVs are primarily small RNA, as you would expect. The EVs are expressing the typical protein and tetraspanin content, as well as the localization. And finally, we did a bioactivity assay, which is in a wound healing scratch test, and we did find that the EV population coming out of the 3D suspension bioreactor systems have the same bioactivity as those from the 2D adherent culture. So then we wanted to test as well how this process works for umbilical cord derived MSCs as opposed to bone marrow, and we found that it was a very similar cell growth kinetics and EV harvest kinetics. So now we've developed a process where the cells at the end of the growth phase are high quality MSCs. We have a reproducible process to harvest EVs in a scalable manufacturing platform that we can scale up. And so here you see that depicted. If you start with the scale down or R&D level, you end up with EVs from 30 to 50 million cells. And as you go from the 3 to the 15 liter to the 50 liter process that have all been shown to show consistent cell yields, that you end up with EVs, EV yields that scale linearly with the volume. So we have this nice consistent CMC process for the generation of EVs. However, the 15 liter scale is not likely to generate the hundreds of EV doses that we would want per lot. So there is still some work to be done in upstream processing in order to increase the EV yield. And of course, there are continued concerns in the downstream process in terms of retention of the EV that is generated from the upstream process. But as we continue to develop our manufacturing processes, it is best to think about EV productivity from various angles. First, to think about biological productivity. And this is EV per cell. And what we found here is that this is a productivity metric that outlines the biological aspects of EV production. This parameter is intrinsic to the cell type and to the culture conditions. And in our studies, we found that, in fact, the biological productivity of the MSCs in terms of generating EVs is twice as high in the 3D bioreactor culture as it is in the 2D flask culture. A second EV productivity that's relevant for manufacturing is process efficiency. When we think about the EV yield per day, this has implications in terms of facility or GMP suite time allocation, which can be very costly. And so this productivity metric highlights the process efficiency of EV production. This parameter allows for comparison of different culture and manufacturing processes for EV generation. And finally, the last consideration in EV productivity is a cost efficiency. And here the units we've developed are EV per liter of media. We have shown and presented elsewhere that the cost of media is the largest driver in the cost of manufacturing MSCs. And therefore, since the large number of EVs are generated by a large number of MSCs, therefore a productivity metric that reflects EV concentration is very important. And this COGS parameter, or cost of goods parameter, reflects the volume of media required to generate a fixed EV yield. And so here you can see that there is a huge benefit from the 3D bioreactor culture compared to the 2D flask culture, again, on the order of a 4-fold increase. And this can be further optimized as we further optimize the bioreactor culture for EV productivity. And thus, I'd like you to consider EV productivity as relevant for EV manufacturing from these 3 points of view: biological productivity, process efficiency, and cost efficiency. And therefore, today I wanted to sum up what we showed you today, which is that we showed that in a bioreactor process that is linearly scalable by volume, such that the 15-liter bioreactor has 5 times the yield of the 3-liter bioreactor, that we have developed a scalable EV manufacturing platform that has increased biological EV productivity and increased cost efficiency. And with that, I'll leave you and thank you for your attention.